In the name of God, last time we talked about infectious disease immunology up to this point, up to this point. So I explained different mechanisms which are involved in defense against intracellular bacteria. And when we say intracellular bacteria or intracellular microbes, we should keep in mind that we have two locations inside the cell. We have phagosomes, and when we say intracellular bacteria or intracellular microbes, we usually mean the ones which are located inside the vesicles, unless specified otherwise. The second group of pathogens which live inside cells are the ones which live inside cytoplasm, and the viruses are basically the major group that do that. So the first step of defense against viruses and other intercellular pathogens which live inside cytoplasm is the production of the so-called interferons. So we had interferon gamma in one of the previous slides when an antigen presenting cell tries to uh, sort of activate a T cell, and then that T cell tries to induce the production of reactive oxygen species. Interferon gamma is involved in that process. But these interferons are slightly different. These interferons are produced by the majority of the cells, like epithelial cells as well as fibroblasts, and pretty much many other cells can produce them too. And when they are produced, they induce sort of a state in the cell, which some people call it antiviral state. So the cell in the middle is infected with a virus. Imagine that these are three liver cells, hepatocytes, and we have a hepatitis virus in here. So the cell in the middle, which is infected, starts producing interferons alpha and beta. And these interferons start influencing nearby cells in a paracrine manner, in a paracrine manner, yes, adjacent cells, and it also influences the cell which has produced it in an autocrine manner. So we have paracrine and autocrine fashions of cytokine action in here. And they generate a situation in which it's very difficult for viruses to replicate. So what they do, well, there are a variety of mechanisms which are activated by interferons alpha and beta. Probably the most important is to decrease the activity of protein synthesis machinery. So cells have a machinery inside them that comprises tRNA molecules and amino acids and ribosomes and the rest of the molecules that enables them to produce proteins. That's one of the major aspects of cell biology, protein production. And viruses go inside cells to use that machinery because they don't have ribosomes and tRNAs and amino acids of their own. So what interferons do, especially interferon alpha and beta, is to decrease the activity of the protein synthesis machinery. So it makes it very difficult for the viruses to replicate. In a sense, if we want to compare them with the human beings or anthropomorphize them, the cell in the middle tells the cells nearby that, well, there is a virus in here, I'm infected. So be careful, decrease your protein synthesis activity so that it makes it difficult for the virus to replicate inside you. And the same cell does the same thing. So this is the very first step of defense against viruses. And it's an innate defense. It's not an adaptive defense. It's an innate mechanism which basically provides like a first line of defense against intracytoplasmic pathogens, interferon response against viruses and the rest of the intracytoplasmic pathogens. The next step is to put sort of the proteins and antigens or peptides derived from protein antigens of the viruses at the surface of MHC class 1. So in the reference book, you probably read about MHC production inside the cells and different ways that peptides get loaded onto MHC class 1 and MHC class 2. So if you have a peptide which is inside the phagosome, it's been phagocytosed, and you have it inside the phagosome, and lysosomes have sort of merged with that phagosome, cutting that protein into small parts. Those peptides will be loaded at the surface of MHC class 2. But if you have a peptide inside cytoplasm, or if you have a protein inside cytoplasm, that gets degraded by a small machinery which is called proteasome. And then the peptides which are produced by proteasome get loaded at the surface of MHC class 1. And the fate of the cell which presents a peptide at the surface of MHC class 2 or MHC class 1 is going to be very different. If it's at the surface of MHC class 2, which happens for antigen presenting cells, CD4 T cells will be attracted. If it's at the surface of MHC class 1, CD8 T cells will be attracted. So these cells contain vesicles which have toxic elements inside them, like perforin and granzyme A. And they also have other mechanisms for killing of the target cell. Exactly, exactly. So we have two sets of mechanisms, releasing the granules, which are cytotoxic granules, and also expressing molecules which can bind to relevant receptors at the surface of the target cell. And overall, they will lead to the apoptosis of the target cell. And the viruses inside, which are in different stages of replication, will be basically eliminated as well. So that's one important mechanism, activation of CD4, CD8 T cells, and cytotoxic activity of these cells will eliminate the infected cell. The point is that some viruses have developed ways to protect themselves against this process. So some viruses during their evolution have learned ways to block different immune mechanisms that we have. And I will talk about it in the second half of the class in more detail. One of those mechanisms is to decrease the production of MHC class 1. One thing that you need, exactly, downregulation. One thing that you need to keep in mind is that these schematic figures that I'm showing you are not drawn to scale, SCALE. They are not drawn to scale. So cell is quite big and molecules are quite small. And if I wanted to draw these things in realistic scales, this would be a very, very tiny entity in here. And it's not going to be only one MHC molecule with one peptide with one receptor. You have to have hundreds of those MHC molecules with hundreds of peptides at their surface, engaging hundreds of receptors at the surface of T cell so that enough stimulation is transmitted to the T cell. So imagine that I need a couple of hundred MHC molecules with the peptide at their surface. If I can decrease the number of MHC molecules at the surface of the infected cell, I can inhibit the process of T cell activation. And that is exactly what viruses do. So they block the expression of MHC class 1, or they decrease the expression of MHC class 1. And that precludes cytotoxic T cells from performing their function because they do not find sufficient number of MHC molecules in order to engage with the target cell. So we have a very important phenomenon during evolution, and it's called evolutionary arms race. What is arms race? Like two rival countries or two enemies which are involved in developing different weapons. So one develops a weapon, the other one develops a counter weapon for that, and they keep going. That is called arms race, like between Americans and Soviets during the Cold War. So evolutionary arms race is sort of an arms race that happens between different organisms which have relations between them, like organisms that have a predator-prey relationship. What is a predator-prey relationship? Organisms that have predator-prey relationship are usually involved in an arms race. The predator develops ways to basically catch the prey faster and better, and the prey also develops ways to escape. So an evolutionary arms race still also exists between host cells or host organisms and microbes. Hosts try to develop ways to defend themselves. Microbes try to develop ways to circumvent that defense. And again, this process goes on and on and on. And basically, if now at the current era in biology, you see that there is a host and there is a microbe, they both exist, it means that the evolutionary arms race up to this point has been a draw. So none of them has succeeded, because if one of them had succeeded, it would have eliminated the other one. All right, and that is something that has happened during the history of evolution. There have been microbes that have eradicated or eliminated the host entirely, or the other way, hosts that have developed ways that the microbes have not been able to circumvent, and they are not getting infected by that microbe anymore. So this is an example of evolutionary arms race. Our mechanism from the microbe side, our mechanism for killing virus infected cells was detecting MHC molecules with the peptide and lysing the target cell. So now some viruses have come up with a way to downregulate the number of MHC molecules. And in that way, they have protected themselves from the cytotoxic activity of T cells. So what has been the countermeasure from our side? What has been the countermeasure from our side? The countermeasure from our side again comes from NK cells. So in the previous example for intracellular intraphagosomal bacteria, we saw that NK cells are involved in the process of macrophage activation by production of interferon gamma, even in the absence of CD4 T cells. Here again, NK cells help by a different mechanism. Exactly, they detect the downregulation of MHC. So NK cells are part of the innate immune system, so they do not have receptors for antigen. They cannot detect antigen at the surface of MHC molecules or in a free form, but they have the ability to detect the presence of MHC molecules, MHC class 1. And the receptor which does that is an inhibitory receptor. Most of the receptors that we know are stimulatory receptors, meaning that when they detect their ligand, they activate the cell. This is completely opposite to that. Inhibitory receptors, when they detect their ligand, the cell is inactivated. It doesn't perform any function. So NK cells always check the presence of somatic cells for the presence of MHC class 1 molecules. If sufficient number of MHC class 1 molecules exist, they get inhibited. But if sufficient number of MHC class 1 molecules do not exist, then they become sort of cautious. Okay, there is probably something wrong with this cell, and we have this ability to kill the cells which do not have MHC class 1 molecules. Nonetheless, because there are some cells in the body which don't have high levels of MHC molecules in their normal state, and those cells are innocent cells, they should not be targeted by NK cells. There is a second check that the NK cell does in order to make sure that the cell that it's facing now and it has low MHC molecules is really a cell in trouble. And it's the presence of the so-called stress antigens. We have a group of proteins which are usually located inside cells. These are self molecules. They don't come from bacteria or viruses. They are not foreign entities. They are self molecules, but they are usually localized inside cells. If a cell undergoes a stress, physical stress, chemical stress, microbial stress, these stress antigens start showing themselves at the surface of the cell. And NK cells have a receptor for them as well. This is a stimulatory receptor, meaning that if it detects the target antigen, it will stimulate the cell. So if I lack sufficient number of MHC molecules at the surface of a cell, which infected cell or some other cell type that has lost its expression of MHC molecules for whatever reason, and at the same time I have the presence of stress antigens, then the NK cells get fully activated. It's like the T cells. The T cells required MHC plus antigen as well as the co-stimulatory molecule to become fully activated. NK cells require the absence of MHC class 1 molecule as well as the presence of the stress antigen. If you have this receptor not seeing its target and this receptor seeing its target, then the NK cell gets fully activated and releases its toxic granules and lyses the target cell. So this is our countermeasure for that. This is our part of the evolutionary arms race. And the interesting thing is that there are again some pathogens which have developed ways to circumvent even this measure. And I will tell you about that. Okay. So we have three mechanisms. One other thing that we need to keep in mind is that both the CD8 T cells that engage MHC molecules and the NK cells that approach these cells because of the lack of sufficient MHC molecule expression produce interferon gamma. And the role of the interferon gamma is to increase the production of MHC class 1 molecule. Because at the beginning, when a virus infected cell presents viral antigens at the surface of MHC class 1, the NK cell comes and engages those MHC antigen molecules. but the sufficient number might not be provided. So it needs a higher number of MHC plus antigen. So it produces interferon gamma. If the cell responds by increasing the MHC molecules and the antigen at the surface, then CD8 T cell can engage the cell and target it. But if the process doesn't work, then the NK cells will approach and say, okay, we gave you a second chance. We introduced interferon gamma for you, but you're not increasing your MHC molecules, so there is something wrong with you. At the same time, you have stress antigens, so the cell that lacks sufficient number of MHC molecules, even in the presence of interferon gamma, as well as showing its stress antigens, is a cell in trouble, and it needs to be basically removed by my cytotoxic mechanisms. So these are mechanisms of defense. The first one, interferon response. Is this an innate or an adaptive mechanism, Mr. Rezai? Is this an innate or an adaptive mechanism? Interferon response. Is this an innate or an adaptive mechanism? Yes. Is this an innate or an adaptive mechanism? This is an innate mechanism. Adaptive mechanisms are the ones that involve B cells or T cells. Then we have CD8 lysis. Is this an innate or an adaptive? This is an adaptive mechanism. Then we have NK cell lysis. Is this an innate or an adaptive mechanism? It's an innate mechanism. So we have three different mechanisms for basically protecting cells from viruses, either by inhibiting their replication through interferon response or by removing the cell that is producing new viruses, either through CD8 T cells or NK T cells or NK cells. So this is the summary of the first part of the class. So we have phagolysosome formation. This is intravesicular. Is this an innate or an adaptive mechanism? Phagolysosome formation, Mr. Rezai. Is this innate or adaptive? It's an innate mechanism. Then we have NK cells activating macrophages. So M5 here means macrophage. Is this an innate or an adaptive mechanism? Again, innate mechanism. And then we have CD4 T helper 1 cells activating macrophages, innate or adaptive. This is adaptive. For intracytosolic ones, we have interferon response, innate or adaptive. It's innate mechanism. CD8 T cell lysis, adaptive, and NK cell lysis, innate mechanism. All right.